All right, so far when we've discussed the endocrine system, we've spent a lot of time talking about the hypothalamic pituitary axis, and we've seen how the hypothalamus helps regulate the pituitary, and then the pituitary will often regulate other endocrine organs. Now we're going to actually look at an organ that is actually outside that HP axis. We're going to look at one that is regulated from a humoral standpoint. In other words, look at the body humors, the body fluids, specifically blood, regulating blood. In fact, we're going to look at the main hormone that's going to regulate, not the only, but the primary one that's going to regulate the all-important solute in your blood plasma, glucose, blood sugar. How are we going to regulate blood sugar? And that's going to come from the pancreas. Now, the other kind of interesting thing about the pancreas is that we're not just going to talk about it right now in the endocrine system, but we're also going to talk about it again in the digestive system. Because here's one great example of where we have a gland that is both an endocrine gland, because it secretes a hormone, and an exocrine gland because, well, it secretes products via ducts to the outside of the body. Well, in this case, the outside of the body is the actual lumen, the inside of the tube of the digestive tract. So I'm going to introduce a little bit of its exocrine digestive functions right now, just to kind of give you a little taste. We'll get back to it when we get to the digestive system. But of course, the primary emphasis right now is to talk about the pancreas and its endocrine functions. So let's just jump right to it. All right. Well, I already kind of give you a little hint that the pancreas is, in fact, has to be near the GI tract, right? In fact, it lies between the inferior border of the stomach, so in other words, just below the stomach, and the proximal portion of the small intestine, which is known as the duodenum. And again, as I've already mentioned, it's both an endocrine gland and an exocrine gland, so they're each going to have their unique type of cells. So we will have both exocrine and endocrine cells. Now, if we look first at the exocrine, just to introduce it, we're going to find out the vast majority of cells in the pancreas are, in fact, exocrine cells. In fact, these special cells are known as pancreatic acini, A-C-I-N-I. And in fact, that makes up about 99% of the pancreas are these exocrine pancreatic acini cells. And of course, by definition, they're going to have ducts, which eventually is going to all kind of come together, sort of like smaller streams going into a big river, into what's known as the pancreatic duct. I'm going to kind of look at the image. I'm going to raise the picture up a little bit so you can see it. The pancreatic duct. And the pancreatic duct is going to merge eventually with the common bile duct, or right next to it at least, and it's going to secrete its contents, I'm going to move this a little more, into the duodenum once again, which is the first part of the small intestine. That being said, okay, again, this is this exocrine function. This has to do with digestion. So what, in fact, is it secreting, you might ask? It's actually going to be secreting an alkaline, enzyme-rich fluid. So it's going to be secreting proteins, specifically proteins, enzymes that are important for digestion. So to give you, once again, just kind of a little hint of how this is going to work when we talk about the digestive system, we're going to eat, right? And when we eat, what do? We chew, we break down our food particles, we fragment it. It's called mechanical digestion into smaller particles, so there's more surface area. So we're breaking kind of the fragments of our carbohydrates, our fats, and our proteins, and so forth. And then it goes into the stomach where there's acid in there. And the acid is going to help denature proteins and kind of also break down these things a little bit more into smaller and smaller components. But it's a very acidic, now we call it chyme. And then from the stomach, it's going to go just a little bit at a time because it's so acidic into the small intestine. And so what's going to happen is we're going to have these enzymes secreted from the pancreas that are, one, alkaline. Why are they alkaline? They're basic because they're neutralizing what's coming out of the stomach. Hence, that's why they are alkaline. And their enzymes, well, what are these enzymes going to do? These enzymes are going to chemically break down the food. We've broken down the food quite a bit in the mouth and in the stomach. We have a little chemical digestion in there too, but now we're really chemically breaking it down with these enzymes from the pancreas. What do I mean by chemically breaking it down? We're breaking those proteins into smaller polypeptides until eventually we get them down into individual amino acids. We break down complex carbohydrates into monosaccharides and so forth. All right. And again, how is it going to do it? It's going to be secreted from the pancreas through a network of secretory ducts. All right. That's all well and good. A little mini introduction for what's to come in the future. But let's now focus on the endocrine portion. Well, the cells for the endocrine portion of the pancreas are known as the pancreatic islets. Or better known as, at least in my case, the islets of Langerhans. I just like to say it that way. It's the islets of Langerhans. And there's two main categories of cells. There's four altogether, and I'm going to mention all four right now, but it's really these first two we're going to care about the most. We have first what are known as alpha cells that could produce a hormone named glucagon. And we're going to have beta cells that actually produce insulin. And I'm pretty sure you've heard of insulin before. Now, you can also see two other cell types there. The delta cells are going to produce a peptide hormone that's actually identical to growth hormone inhibiting hormone. And the what's known as F cells are going to secrete pancreatic polypeptide. For right now, we might come back to that later in class, but right now I want to just focus on the alpha cells. Basically, what the delta cells do is they actually suppress the release of glucagon and insulin, and they decrease the rate of food absorption. While the F cells inhibit the gallbladder contraction. Well, you might say, well, what's that? If we look at the image once again, we can see the gallbladder right here. What's going to happen is that we need to be able to separate our fats, and because the fats are going to want to clump up, because they're, of course, fats are hydrophobic. They're all going to want to stick together. We need to kind of break it up so enzymes can actually have more surface area to attack. That's what bile is. Bile is actually produced in the liver and stored and kind of concentrated in the gallbladder. And so when you eat something, you're not just secreting the pancreatic enzymes, you're also secreting bile to kind of break up, the technical term is emulsify fats. Break them up into smaller droplets so the enzymes can actually attack it better, if you will. Well, the F cells will actually inhibit gallbladder contraction, so there's going to be less bile, and actually decrease the production of some pancreatic enzymes. And I apologize, my pen's acting up on me a little bit today. Point being is, we will revisit these, especially when we talk more about digestion. For right now, they are hormones. For right now, let's focus on insulin and glucagon. These two hormones contribute to the minute-by-minute control of blood glucose levels. So, what do I mean by that? Well, when you initially eat something, right, all of a sudden you're breaking down, digesting your carbohydrates, and it's going to be absorbed in the small intestine. That means now we have glucose in the blood, more glucose in the blood. So when blood glucose levels rise, the beta cells of the pancreas are going to secrete insulin. And what that's going to do is going to stimulate the transport of glucose across cell membranes, across the plasma membranes. Basically, it's a peptide hormone, and when it hits its receptor on the cell membrane, it's going to create a chain of events where we're actually going to take a pre-made glucose transporter, a GLUT transporter they'll call it, and that will then go to the plasma membrane to be open to let the glucose in. So it's literally telling cells, let glucose in. There's glucose in the blood, let it in. The main targets that are going to actually be the liver, muscle, and adipose tissue. We'll talk about all of those. Now let's say it's the other way. Let's say instead it's been a long time since you've eaten. Okay, so we actually would call this, to be clear, what I'm referring to now as a fasted state. While the other one would have been, of course, a fed state. So, if you're in a fasted state, it's been a while since you've eaten, then those alpha cells will release glucagon. And, of course, stimulate the release of glucose, especially by the liver. Now we're going to find out, you already know that the body stores glucose in the form of glycogen. It's kind of a metabolically inactive form in both the muscle and the liver. For the most part, the glycogen in the muscle is really going to be for the muscle right there. The glycogen in the liver is to maintain blood glucose levels because that's what the brain wants to work on, okay? The brain cannot run on fats, it cannot run on proteins. It can run on glucose, blood sugar. It can also run on ketones, and that's what happens. You may have heard of a ketonic diet. You actually build ketones in the blood, called going through ketoacidosis. When we talk later on about nutrition, we'll come back to that. But the brain only works on glucose. That's its primary one, and as secondary as a backup, if you're not getting enough carbohydrates, it can run on ketones, which are a byproduct of fat metabolism. Okay, so let's now get into a little bit more specifics. Let me zoom in a little this time. Okay, so as I already mentioned, insulin is a peptide hormone. And we know it's released by beta cells. As far as its target cells, the liver, adipose tissue, muscles, and so forth, is again, it's going to accelerate glucose uptake. That's its job. And again, it's going to do so by increasing what we call the GLUT transporters in the plasma membrane. All right, what else? It's going to accelerate glucose utilization and enhance ATP production. So it's actually going to kick up metabolism a little bit. It's also going to be, well, let's break things down a little bit. Say we're using energy, that's great. So the glucose comes in, we're using it right away, but it's also going to go to storage. So let's say blood sugar is plenty high. That's why insulin's being produced. So it's going to, knock on the liver's door and say, hey, this is it from the islets of Langerhans. We have insulin here, right? And hey, can you let some glucose in? And when that happens, we have glycogen formation. And that'll happen again both in the liver and in the muscle as well. However, I'm kind of skipping a bullet point. If glycogen stores are basically filled in the liver and in the muscle, then what it'll do is it'll go to that adipose tissue and it'll actually stimulate triglyceride formation. In other words, we start building up fat. Notice this is also going to help as far as taking up amino acids as well, because if you think about it, you don't usually eat just a carbohydrate, pure carbohydrate, not like you just take big spoonfuls of sugar typically. When you eat things, it's usually a mixture of carbohydrates, proteins, and fats, and what have you. So when you're taking a carbohydrate, your blood sugar goes up, it's going to secrete insulin. Insulin's not just going to help the uptake of glucose, it's also going to help with the uptake of amino acids. Again, glucagon is released by the alpha cells. And what they're going to do is they're going to mobilize energy reserves. It's going to stimulate the breakdown of glycogen in skeletal muscles and liver. Let me raise this up a little bit. So it's going to be. So we're going to break down that stored form of glucose to then release it into the bloodstream. Likewise, it'll also stimulate the breakdown of triglycerides in adipose tissue. And then what we'll also do is it's going to try to produce glucose to raise glucose any way we can. So it'll actually use gluconeogenesis in the liver. It's kind of the opposite pathway of glycolysis, if you will. All right, so now let's think about what normally happens here. Now, the key numbers to know, this is one of the few that I really kind of like you to know, are 80 and 100. So it has it, and this particular graphic is homeostasis is between 70 and 110. It depends on what book you read, quite frankly. Milligrams of glucose per deciliter of blood. Okay, so this graphic has 70 to 110. I usually hear 80 to 100. Okay, so what happens? Let's say you haven't eaten in a while, so you are in a fasted state. And you have low blood sugar. So if you were to take a sample of your blood and monitor it, say you're at a 50. Well, you know what's going to happen now. You have glucagon released from the alpha cells into the bloodstream. We know that glucagon is actually inhibiting cells, slowing them down the metabolism, so they're not going to be using as much glucose, but it's going to stimulate the breakdown of glycolysis. The breakdown of glycogen, which is glycogenolysis. It's the breakdown of glycogen into glucose, so release glucose in the blood. It's also going to stimulate, as I already mentioned, gluconeogenesis, so we can actually make glucose, say, from amino acids, from lactate, free glycerol. All these things can, the glycerol would be from triglycerides, right? And we can actually make glucose from these precursors. All of this is to actually raise blood glucose levels back to homeostasis. So that's when we're in a fasted state and blood glucose levels are too low. We go up here, we are in a fed state. We just ate, we check our blood glucose, and it's now 160. What's going to happen? Okay, we need to bring the blood glucose down. Insulin's going to be released from the beta cells of the pancreas, and it's going to go to those same areas, right? What do we see? We're actually going to increase metabolism a little bit. We're going to inhibit glycogenolysis. In other words, we don't want to break down glycogen anymore. In fact, we want to have glycogenesis, in that we're going to actually make glycogen, and it's going to inhibit gluconeogenesis. So once again, the opposite effects. And it's going to tell the cell membranes to create, once again, the glucose, or actually move the GLUT transporter to the plasma membranes, so they will let glucose into the cell. All right. So next time what we're going to do is we're going to talk briefly about diabetes mellitus. We'll talk a little bit more about it when we talk about other pathologies. We're going to talk about this one twice because it's so important.